Hello and welcome to the second episode of the new Innova Biosciences webinar series. This webinar is in association with iPro Interactive and GE Healthcare Life Sciences. Today's webinar, A Beginner's Guide to Lateral Flow Assay Development, will introduce you to various aspects of lateral flow assays including antibody, antigen and material selection, basic troubleshooting and the covalent attachment of antibodies and ligands to gold nanoparticles. My name is Claudina and I will briefly introduce our three speakers and explain the structure of this talk. Today, Dr. Ahmed Jahanli from iPro Interactive, Dr. Klaus Hochleitner from GE Healthcare Life Sciences, and Tom Speedy from Innova Biosciences take you through our Beginner's Guide to Lateral Flow Assay Development. This webinar is divided into three parts. First, Ahmed, the Director of Research and Development from iPro Interactive, will tell you about antibody and antigen selection for lateral flow tests. iPro Interactive is a company specializing in the development of quantitative lateral flow tests for analytes in saliva with applications mainly in the field of sports medicine. iPro also offers contract research and development services. Klaus, the Global Lead Technology Product Specialist from GE Healthcare Life Sciences, will discuss material selection, material properties, and basic troubleshooting for lateral flow rapid tests. GE Healthcare Life Sciences provides integrated components, platforms, and technology solutions for point-of-care immunodiagnostics and molecular diagnostics. Tom, our Corporate Business Manager from Innova Biosciences, will introduce you to the covalent attachment of antibodies and ligands to gold nanoparticles. Innova Biosciences specializes in bioconjugation technologies.
Hello, can everyone hear me? I think you are. Yes, I'll just repeat again. Welcome everyone in case I was muted. I'll start my talk now and with the introduction. Lateral flow assay development is driven mainly by the need to move some diagnostic immunoassays from the laboratory setting to a point of contact or point of care setting. In reality, the expansion of lateral flow assays started with the development of pregnancy tests in the 80s by Unipath, and that opened the gates kind of for the development of assays in other regions, areas as well. The expansion now spreads across many biological fields, not only in clinical medicine, but in veterinary medicine and environmental testing and defense and so on. And I believe that with improvement in reproducibility, sensitivity, quantitation, and connectivity, lateral flow assays will see massive increase on a very huge market in the very near future. Sorry. The basic components of a lateral flow test involves these five components, which are membrane strips, test site, sample pad, conjugate, absorbent, and the antigen antibody reagents. The next speaker, Klaus, will talk about the first 4 points, and I'll be only addressing the last one. Lateral flow test types tend to be of two types. The first one is the sandwich assay format, which is normally used for large analytes, like proteins with multiple antigenic determinants. Competitive assay formats are used for small molecules like hormones, drugs, antibiotics. This is just an illustration of a sandwich lateral flow assay. On the left-hand side, this is what's termed a double antibody sandwich for antigen detection. And in this case, we have one antibody immobilized on the test membrane, which captures the analyte, and then the detector molecule antibody forms a sandwich with the immobilized antibody. The detector molecule could be a gold particle, latex particle, a fluorescent chromogen, or a biotin or an avidin system. There's also another example of a sandwich assay. In this case, this one is used for detecting antibodies to, say, bacterial antigens. And in this case, the sandwich is formed by immobilized antigen and the detector antibody. With this sandwich assays, the signal that one obtains is directly proportional to the concentration of the analyte in the test sample. The second format of lateral flow assays is the competitive inhibition, which is used for assaying for small molecules, because small molecules cannot cater or bind more than one antibody. And in this case, the small molecule or the analyte is immobilized by binding it first to a carrier protein like bovine serum albumin, and that's immobilized on the membrane, and then you have the antibody to the analyte conjugated with the detector molecule. In this case, the signal is inversely proportional to the concentration of the analyte in the sample test. Now we come to the choice of reagents. First one, the most important reagent is the antibodies. We have two possibilities, which are either using polyclonal or monoclonal antibodies. Polyclonal antibodies have the advantage of it's quite easy to obtain high affinity antibodies. There's a wide choice of species to raise the antibodies in them. The disadvantages are the immunizing antigen has to be very pure, otherwise you have a problem with specificity. Second problem or disadvantage is that out of the total immunoglobulin fraction in the antiserum, one would be lucky to get more than 5% as the pure antibody to the analyte in question. So therefore, immunoaffinity purification is very essential. And the final point is that the supply is limited and it's related to how long the immunized animal lasts. Monoclonal antibodies have the advantages. There's an unlimited supply once you get the hybridoma. So you have theoretically an unlimited supply of the antibody. You can get high specificity by doing the selection, so even the antigen doesn't have to be pure. And finally, immunoaffinity purification is not necessary. The main disadvantage of monoclonal antibodies is it could be quite difficult sometimes to get high affinity antibodies because of the lack of polyclonality. And also there's a limited choice of species apart from using mouse or murine monoclonal antibodies. Now the source of antibodies are antigens. If one is planning to develop a lateral flow assay using existing antigen-antibody combination, then there's many commercial sources of such antibodies. I would recommend that people use antibody databases like biocompare.com or Linscott directory to search for the required antibody and antigen. There are many antigen-antibody pairings available in the market, especially for cardiac markers, steroids, and drugs of abuse. And many of these are actually designed for lateral flow assays. Whatever the source of the antibody or the antigen, one must ensure that the supplier can guarantee or secure continuous supply of the reagents. The other alternative is to make the antibodies and antigens in-house, and that's basically what would happen if someone is developing a new lateral flow assay or a new immunoassay. And again, there are many companies that provide contract immunization, both for producing polyclonal antibodies and monoclonal antibodies, so one doesn't have to worry about establishing animal houses and facilities. For polyclonal antibodies, there's a very wide choice of species one can use. Recently, many people say that chicken antibodies would be very good to use because they lack complement fixation. But however, there are other problems that can be involved with using chicken antibodies. For monoclonal antibodies, murine or mouse antibodies are the first choice because they've been in existence for over 30 years, well-established technology. But there are other species antibodies like rabbit monoclonals that can be obtained from Abcam, a company based here in Britain. There's another company that produces sheep monoclonal antibodies, Bioventix, also in Britain. And the other choice is recombinant and engineered antibodies, but these are not really widely commercially available and tend to be very costly. And also I need to mention that for monoclonal antibodies from other species, they tend to be protected by patents, and there will always be a possibility of having to pay royalty for using these reagents commercially. Now, in terms of immunization, depending on the type of antigen, if one is looking for proteins, then in general, anything above 5,000 in molecular weight can be used for immunization, of course, coupled with using adjuvants. But if the antigen, even protein, is not highly immunogenic, which could happen if someone is trying to look at proteins with very low differences in amino acids between species, then it's possible to raise antibodies to such a molecule by haptenizing the molecule by just coupling it to KLH. An example of this is making antibodies to platelet-derived growth factor, which is a 28,000 molecular weight protein, which is highly basic and very difficult to make antibodies to. For small molecules, it is important that the molecules are coupled covalently to a carrier protein. Otherwise, you won't get any immune response to the small molecules. And in this case, it's very important to look at the ratio of hapten to proteins, because that could have an impact on the affinity of the antibody raise. And many of the rules involved here, or protocols, are very empirical. There's no set rules as such, and quite often you need to do it by trial and error. The choice of spacer and linking the small molecule to the carrier protein is very important, because by using a large spacer, it's possible to make antibodies that will recognize the spacer rather than the hapten, or a combination of the hapten and the spacer. My preference is to use carbodiamide if that is possible, or if you have a carboxylate group in your molecule, because there's hardly any spacer and the antibodies can almost be specific for the hapten. Alternatively, one can use a different spacer for immunization and for the assay. I would suggest that people avoid using bovine serum albumin or ovine albumin, because both of these are commonly used in lateral flow assays for blocking and for removing non-specific signal. Once the antibodies are obtained, then the next step is to do evaluation of the assay. And I would hardly recommend that people don't move straight into developing a lateral flow assay, and first to evaluate the antibodies on the actual assay using a laboratory-based system. And because you could also have enzyme immunoassays based on the sandwich or the competitive inhibition format, so that kind of mimics what you get in the lateral flow assay as well. But one has to keep in mind that it is not a direct correlation between the lab-based assay and the lateral flow. The lab-based assay, especially those done in microtiter plates, I would define them as being assays in a closed system, because you have everything there in the microtiter well. While with the lateral flow assays, it's like what I term as an open system, because you have a continuous flow of reagents on top of the capture molecule and so on. So it is not a direct follow-up or correlation between the lab test and the lateral flow. One needs to keep that in mind. And also, I think it's very important to almost quickly to move into looking at your analyte and the matrix that the assay is designed for, and not to dwell too much on doing work testing the analyte in buffers, because you could waste a lot of time doing that, and you find many problems when it comes to doing the lateral flow assay. The major issues with the lateral flow assays are really two, basically: sensitivity and getting rid of the non-specific signal that quite often happens in lateral flow assays. I mean, this presentation, there's not enough time to go through all the different options that one can use. I've just listed some here, like with the gold because that's what I myself use gold antibody conjugates. One can increase or the sensitivity and reduce the non-specific signal by controlling the amount of antibody bound to the gold particles. The other option is to do a signal amplification by using a bioconjugation system. You can control the sensitivity by the amount of reagents deposited on the membrane. And also the last point is the sample and buffer components. These can be either buffers used for running the test or buffer components included in the sample pad. And there are many, many different modifications that one can introduce into this to try and reduce the non-specific binding. For instance, if your assay requires a very high level of detergent or a very strong detergents that are like anionic or cationic detergents, then it might be advisable to use a covalently bound antibody to the gold particles or to the, so that you can use a high concentration of detergents without the possibility of dissociating the antibody from the gold particles. Other aspects of using the buffer, including proteins, different types of proteins in the run buffer or in the sample pad to try and reduce non-specific binding. And of course, there are the other option of how you treat the membranes, which Klaus, I'm sure, will be dealing with that. Just to conclude, really, the two main important aspects of lateral flow assay is the quality of the antibody and the antigen used in the system. The more specific the antibody is, the more likely that the assay will be sensitive and that you will be reducing the amount of non-specific signal that you might get in. Of course, that means the antibody affinity has to be a priority, especially if one is developing a very sensitive type of assays. The purity of the antigens is also very important to avoid getting cross-reactivity with unrelated analytes. And also in the case of looking at small molecules, the type of antigen or the type of hapten carrier molecule one uses would have an impact as well in terms of the substitution ratio of the hapten molecule with the carrier protein. It can have an impact on the sensitivity of the assay, especially in competitive inhibition assays. And I think that concludes my presentation, and I look forward to receiving questions from the attendees. And thank you very much for listening.
Okay, welcome to this webinar. Sorry for the confusion. I had some issues with my computer. Well, what you see here is what you probably have seen several times. This is the typical rapid test, the lateral flow device, and it is consisting basically of 4 different materials that fulfill different functions. So the sample at first sees the sample pad that prepares the sample for running through the test strip. Then it meets the conjugate pad, where it will find a dry detector reagent that hopefully finds the target, and then moves on to the membrane, which is the only part of the test that the end user might see. And finally will be, the target will be bound on what we call a test line, and there will be a control line that tells you that the test is running and that the conjugate was physically intact, and that you probably can rely on the test result even if you have a negative one. And finally, the sample sees an absorbent paper that soaks through the liquid and keeps it there until the test is read out. I now would like to go through all of these materials in the order the sample sees them. So what about the sample pad? What do you have to do before you can select the sample pad? Well, the first thing that you have to do is you have to think about the variability of your sample as such. At first, What is the number of target molecules you can detect and won't detect with your antibodies and your test system at minimum? What is the maximum number of target molecules that you can expect in a sample under reasonable conditions? And this defines the sample volume that is to be applied. The second question is, what is the variability of the sample composition in terms, for example, pH and salt content? The problem here is that the animal in which you have raised your antibodies has optimized the antibodies for you at a pH of 7.4 and an ionic strength that is more or less the one that you call PBS, phosphate buffered saline. The live sample from the patient can vary a lot in terms of pH, in terms of salt, of lipid content. If you think about saliva, even the same sample from the same person can vary a lot depending on the time of day, nutritional state, and the like. And that means what you have to do is you have to adjust your sample liquid in the sample pad to a sort of common conditions so that in every test you run, the detector antibodies and the test line antibodies see always the same sample composition in terms of pH, salt, and the like, so that you have reproducible results and comparable results. Next problem is sample viscosity. The more viscous the sample, the more open as the pad material has to be. And what you need to know is, do you have unspecific interactions of the target molecule with the pad material, because that defines whether and how you will block the sample pad. And are there unspecific interactions of the target with test line reagents? In that case, we talk about false positive signals. And it might be that this will lead to additional adjustment requirements of the sample pad. And finally, there might be tests where you would like to retain particles that are part of the sample liquid. That can be resolubilized fecal material, that can be resolubilized foodstuff, but the most prominent particle that is to be retained in sample pads is red blood cells. With respect to selection, after you have specified the sample volume that you would like to apply to the strip, you ask your provider for the absorption capacity of possible sample pads, and you will get a value in microliters per square centimeters, a comment on the paper raw material, and perhaps the presence of binders. Binders are polymers that stabilize papers and glass fiber materials. Usually we talk about things like polyphenolic alcohol or polyphenol pyrrolidone, and they will inevitably become part of your test system because they will not stay in the pad. A bit of it will leak out and will migrate further on into the conjugate pad and onto the membrane. The general advice is select high-quality chromatography papers as sample pads. Select cotton linters papers. Cotton linters is a very reproducible raw material for pads, whereas wood pulp may come from this wood today and from another tree tomorrow, and this can lead to difficulties in reproducibility of the sample pad properties. If the sample pad is to retain particles, especially when we talk about red blood cells, it might be that you would like to run with one pad that serves as sample pad as conjugate pad. At that point, you are bound to glass fiber pad materials. So what you then do with the sample pad is you will pre-treat it with an appropriate buffer system. Usually you will do that by immersion. So you soak the sample pad and dry it. Please dry the material in a forced air convection oven. What you have to do is you have to guarantee that all material is dry. That means it's not over-dried, but it's also not humid anymore. Any inconsistency in drying conditions will lead to inconsistency material behavior in the finished rapid test system. After drying the material, please store the coated material at what most people routinely call room temperature and less than 20% relative humidity. Please keep in mind that what you have created is a very hygroscopic material, and that means this material will try to rehumidify. And that means, if you allow that you will slowly get humid materials, which is the perfect breeding ground for all kinds of microbes, and will at least reduce the shelf life of the rapid test that you have manufactured. And this holds true for all coated materials, including coated membranes and lateral flow tests. The pretreatment buffer for a sample pad usually contains a buffer for pH adjustment, and this is always necessary. It may contain salts. Use it at low concentrations if you use salt at all, because the salt will add up to the salt that you have already in your sample, and it will add up to the salt that you may have in test and control line reagents. So you can reach relatively high salt concentrations by that approach. You will probably need blockers like proteins or polymers in order to prevent unspecific interactions, and you will use non-ionic surfactants in order to increase the wettability of the pad material and support blocking. And they may help to reduce unspecific bindings of your target to the test line. If you plan to develop a serum-based lateral flow test, keep in mind that sooner or later somebody may ask you to run the same test with whole blood as a sample. In that case, it's a very wise decision to develop the serum-based test with reagents that are not hemolytic. That means that do not lyse red blood cells. A special word on blood samples. In blood samples, you would like to retain the red blood cells in the sample pad, or in that case, people tend to call this pad a blood separation pad. These cells are always retained by mechanical interaction, as is illustrated in this electron micrograph here, and in the better magnification here. And these cells wrap around fibers. If you go with whole blood samples, you have to define your blood volume very carefully, because it's not only the question how many serum do you need to see a signal that you want to see. The question is also how many blood cells do you apply with that serum, because too much volume will lead to red blood cell breakthrough problems, and this is the background you will not be able to block. And of course, you must avoid hemolysis, because free hemoglobin on your membrane will result in a background color that's difficult to deal with. Signal from red gold particles on a red background gives usually very poor signal-to-noise ratios. The next pad that your sample sees is the conjugate pad. And typically, this is the physically smallest part in a lateral flow test, but you would like to do a number of jobs for you. At first, it must absorb the volume of detector conjugate that you add to the pad. It must not interact with the conjugate. It is to maintain the conjugate integrity upon drying and in the dry state, which can easily be more than a year at what people call room temperature. And what you then expect is that as soon as the sample liquid enters the conjugate pad, this pad releases this conjugate happily into solution in an active form, and the detector antibody that's conjugated to the conjugate will then interact with your target. And this happens quickly, and the conjugate pad does allow for this interaction in terms of the chemical composition of what is then the liquid in the conjugate pad. So you see it's an awful number of functions, and to our experience, most issues with rapid tests or have their origin in the conjugate pad, or mistakes that have been done in pre-treatment of the conjugate pad or in the conjugate itself. What you need to know before you can select a conjugate is, at first, what is the type of conjugate do you want to use? Do we talk about metal colloids like gold colloids, let's say of a diameter of 40 nanometers? Will we talk about latex beads that routinely come in a diameter of around about 300 nanometers? Will you do covalent or non-covalent binding of the detector molecules to the particles, or will you not use any particles at all but go with directly labeled antibodies or antigens? What you have to define is the amount of detector molecules needed in the test in order to obtain the required sensitivity. And this can be very high. The law of mass action also applies to lateral flow tests. And the worst case that you can have is that you run an antibody search assay, for example, for a human antibody specific for a specific target molecule like an HIV or tuberculosis antigen that you have applied to the test line. And you have to deal with the huge excess of antibodies in the human serum that is specific for anything but not for your target, but that will be detected by your detector reagents. What you also have to keep in mind, what is the maximum concentration that can be achieved with the conjugate in solution without inducing aggregation? And this is not only true for the solution that you dispense. You dry the solution. That means during the drying process, you will withdraw water, and you will increase the concentration of your conjugate particles, and they must not aggregate upon drying. And this defines the volume of conjugate and the concentration of conjugate that you must apply. And again, you simply ask for the absorption capacity of the conjugate pads that are available, select the pad material, and you may calculate then the pad size that you need per test. As conjugate pad materials, we routinely talk about either glass fiber pads or nonwovens. Glass fibers are more versatile, especially when it comes to additional pad functions, so a combined pad of sample pad and conjugate pad. And in most cases, people like to work with glass fibers. But you have to keep in mind, if you want to go with reel-to-reel machinery, you will have to select your glass fiber pads very carefully, because they have to have a reasonable mechanical stability even in a wet state. And with glass fibers, that can become critical. What you have to do in terms of pretreatment is a pH adjustment. You always have to run with a sort of buffer system. Our recommendation is do not use salts, especially when we talk about metal colloids with non-covalently bound antibodies. These will not survive high salt concentrations during drying. You will need blockers, and you will need non-ionic surfactants, especially non-ionic surfactants that are able to block membranes, what we call on the fly. The classical example is T20 or Triton X100 as part of the pretreatment buffer of the conjugate pad. It's dissolved by the sample, and it migrates much faster than the conjugate across the membrane surface. And while migrating across the membrane surface, the surfactants bind to protein binding sites on the membrane and then block the membranes while the sample is migrating. And this is the preferred option for blocking a membrane or unspecific binding sites on the membrane, because it simply eliminates a separate manufacturing step that is called blocking the membrane and validating the blocking procedure. A good question always is, how do I get my conjugate into the pad? Basically, there's two options. One is an immersion approach. That means you take the pad, put it into a tray with the conjugate solution, soak the pad with the conjugate solution, and convert it to smaller strips. The second option is dispensing of defined conjugate volumes per conjugate pad length or area. The problem with immersion and dipping is that all pad materials are variable in thickness. Your test strip is limited in terms of width, and you have defined the pad length. So variables in pad thickness will lead to variable amounts of conjugate in the test strips in the soaking process, and that means you will have variable test results, and that may give you issues with respect to test reproducibility. And therefore, we strongly recommend dispense your conjugate in defined volumes per centimeter of pad length, because this is the only way you have to guarantee constant reaction conditions from test to test. The next step is the liquid enters the analytical membrane. Typically, we talk about what people call a large pore size nitrocellulose membranes. Nitrocellulose membranes are available in very broad range of pore sizes, that means some flow characteristics. What you should know is that all NC membranes on the marketplace contain a surfactant, usually an anionic surfactant, that makes them hydrophilic. Every membrane manufacturer uses his own surfactant, so that's proprietary. But at the end of the day, it will become part of your test system, and it will influence your antigen-antibody interaction at test and control lines. The structure of NC membranes is a bit far away from what people call a porous structure, because what an NC membrane is, it's a meshwork of nitrocellulose fibers, as you see here in this electron micrograph. There are no pores, and the best comparison is a sponge. And the inner surface of this spongy structure gives you the high binding capacity that you need for proteins, and that gives you an option at all to construct lateral flow tests with the required protein binding capacity and sensitivity. Membranes are characterized by a parameter that people call capillary flow time or capillary rise time or wicking time. These are the terms that I've used. And all of them describe the same. The time a liquid, and in most cases we talk about water, needs to migrate a defined distance, and at always 4 centimeters, parallel to the membrane surface against gravity. Here at the lower left, you see a typical test setup for that. You have a membrane strip with a width of 10 millimeters and a defined water volume, and this water volume migrates upwards, and what people are interested in, what is the time the liquid needs until it reaches the mark at 4 centimeters. And typically, you then come up with membrane specifications that look like this, which is a new family of membranes that GE has developed for very sensitive and very reproducible tests. And what you see is you have windows in capillary flow times, and that you can use to define your membrane needs. For the membrane selection, the parameters to consider are sample type, the test duration you would like to have, and membrane variability. The bottom line is, the more viscous a sample is, the slower it will run through a membrane. And the slower a membrane is, the more nitrocellulose it contains per square centimeter surface area, which means it binds more protein. The unfortunate thing is, a very sensitive membrane, that means a slow membrane, is unlikely to give you the fastest test in the world, and that means at some point you will have to decide what you want to have, a terribly rapid test or a very sensitive one. Especially for highly sensitive and quantitative tests, use membranes with very low CVs on capillary flow times. As a general recommendation for test development for water and liquids with comparable viscosity, go with a slow membrane. For urine and low blood and serum volumes, with chase buffer gold with a medium fast sample, with a medium fast membrane, and for undiluted serum, saliva and resolubilized solids, the recommendation is use a fast membrane. Reagent quality is an issue here. An extremely high affinity antibody at the test line can make a test working that runs with urine or even low serum volumes on a very fast membrane, and even water can then run on a very fast membrane. A low affinity membrane, a low affinity antibody will probably not work even with undiluted serum on a fast membrane. You might have to go to medium fast or even a slow membrane. The price you have to pay then is a very slow rapid test. What you have to do with the membrane, you have to dispense protein lines on them, a test and a control line, and most reason for concern is the test line because this is that generates a signal. So recommendation is use precision dispensing equipment as early as you can in test development. You may do first feasibility studies just with using an Eppendorf pipette or something comparable, but at the end of the day, only precision equipment will give you the lines that you need to really see whether you have developed a reproducible test system. Typical dispensing rates are varying between 0.6 microliters per centimeter and more than 1 microliters per centimeter. Typical protein concentrations are around 1 microgram per microliter. A general recommendation is apply your proteins to the membrane in a buffer that is close to the protein's isoelectric point. This holds true as long as this pH is not below 3 or above 10, because pHs below 3 and above 10 will start to hydrolyze the cellulose backbone of the membrane, and then you're done and you can't work with the membrane anymore. The dispensing buffer should not contain high salt concentration. Even PBS can be a too high salt concentration, because what happens is that you dry the salt on the test line and you have to re-wet these salts with your sample when it approaches the test line. And it can easily be that this takes more time than you would like it to take. And additionally, it may happen that the liquid, when it leaves the test line, does not run smoothly anymore, but gives you distorted migration fronts that look like a U or a V. The buffer may also contain low concentration of methanol and ethanol that also destabilizes proteins and facilitates protein binding. And please try to avoid the use of surfactants in the dispensing buffer At first, you might see foaming problems. The second problem is surfactants may compete with proteins for binding sites. And sometimes it's advisable to use low concentration of trialules because this may increase the protein stability upon drying. The final material that your sample is seeing is the wick. The task of the wick is to soak the sample liquid completely, and especially all reagents that have not been absorbed somewhere on the membrane. And the most important task is this one. It must prevent the backflow of this liquid into the drying membrane. Please keep in mind that the membrane is the thinnest part of your rapid test. That means it will dry first. Any liquid that flows back from the wick onto the dry membrane will contain unbound conjugate particles. And these conjugate particles will then settle somewhere on the membrane, and they have a tendency to bind very well to formally negative test lines in sandwich assays, making them seemingly positive. And that means that you have to define a readout time window for your rapid test. You must clearly state a time in minutes after sample application when people may have a look to the test and read out the signal, and the time in minutes after sample application, after which it's baseless to read the test. As material selection, the recommendation is relatively simple. Select a constant linters paper with an absorption capacity that is much higher than the sample volume. I would like to finish this presentation with some very basic troubleshooting. So membrane issues that you see as, at first, uneven lines or dots, depending on what exactly you spot to your membrane. And in most cases, we talk about basically the wrong membrane. You need a faster or a slower membrane to get a more even line. You may have too much reagent volume that you dispense on the membrane, and in that case, you reduce the volume and in parallel increase the protein concentration of the reagent that you want to dispense. And what you should look for is for dispensing buffer composition in general, and have a look for your dispensing process. Sometimes it's simply the dispenser that gives you a problem. False positive signals can have a variety of reasons. The most prominent one that I've found is pH. So I have seen many tests where you simply have to had to run at elevated pHs up to 9.5 in order to get rid of ionic interactions at the test line. What also can happen that you have hydrophobic interaction. In that case, surfactant may help. And in some cases, you have, unfortunately, very specific interactions that give you false positive signals. The most prominent example that I've seen was an antigen on the test line that was a lectin and that could interact with Fc parts of conjugated antibodies very well and always gave a faint positive signal regardless of what the sample composition was. And at the end of the day, we switched to an FAB2 fragment that we conjugated, and then the false positive signal was gone. For false negative signal, all these parameters can play a role, but here it might be advisable to use a slower membrane or to increase the sample volume in order to get more target bound, or to give, in case of a slower membrane, the test line reagent more time to grab the target out of the migrating liquid. And a more cosmetic issue is uneven liquid fronts of migrating samples. In that case, discuss with your membrane manufacturer. Is the membrane too old? Do you simply have not enough surfactant in your system? That's a quite frequent reason for uneven liquid fronts, and that means you need more non-ionic surfactant. Or do you run your test at extremely low relative humidities? Because this will always lead to hydrophobicity issues, and this leads to uneven liquid fronts. Well, that's it so far. Whatever you need and whatever you have in terms of questions, please come back to us. And the general advice is do not try to reinvent the wheel, but talk to all of your reagent material suppliers when you run into trouble. We are here to help, and we will help if you let us do that. Thank you.
Hello, and thank you all for joining us today. This is the final section of the webinar discussing covalent attachment of antibodies and ligands to gold nanoparticles. I'll give you a brief overview of Innova Biosciences and our bioconjugation expertise. Sadly, we don't have time to cover the other particles that are commonly used in lateral flow, but I'll touch on this briefly before we move into the gold. So Innova was founded on lightning link technology. Within that range we have many fluorochromes and enzymes, biotin, streptavidin, many of which can be applied to lateral flow. The basis of this is the speed at which you can do the conjugation, the reproducibility and the stability of those conjugates once completed. If anybody has any questions, please we can follow those up later. Here's a brief summary of the methodology. So you simply add your antibody to the lyophilized labeling mixture, incubate, add a quencher, and then your conjugated antibodies are ready to use with the rapid kits in less than 20 minutes. So today we're talking about gold. The most common gold particles used in diagnostics are 20 to 80 nanometers, the most common being 40 nanometers. These have traditionally been conjugated by non-covalent passive absorption technologies or more recently attaching ligands or other biomolecules to the naked gold to try and strengthen that conjugate. The difficulties associated with this are the needs for optimizing the conditions for each different antibody, such as pH, salt concentrations, etc., hitting the correct PI, various centrifusion and other processing that's involved in that. At Innova, we measure the quality and the dispersal of the gold colloid before we apply the coat using this 650, 530 wavelength scan. You can quite clearly see here that when the particles are aggregated, you get this profile and you get this purple color in your gold colloid. When it's properly dispersed in the solution, you get a much clearer peak here, and you get this pinky red color. Self-assembly methods, the use of ligands have been increasingly common. Again, it's an improvement on traditional passive methods, but it's still susceptible to problems. You can get a successful coating, which here you can see it's completely coating the particle, but sadly this can dissociate and then you result in this aggregation that can also be caused by destabilization of that particle. So what we've actually done, here you see the gold and the latex. Again, we won't have time to cover latex today, but we will touch on that. I will come back to that in a future webinar. So we've taken the gold particle and we've applied a surface coat to that, which then allows us to functionalize that, significantly increases the stability of the particle as a pre-conjugate colloidal stability is significantly increased. And we can also control the number of reactive groups, so we can vary that so we can have a high antibody loading or low antibody loading, or using different chemistries as we see on this slide. So these are the main chemistries available for coupling to the particle. With the Innova coat, we bind to the amines as a standard, but we can also adapt or provide custom gold conjugation kits with these other chemistries as well. So here you have an example of the enhanced colloidal stability at relatively low concentration conditions, such as sodium hydroxide, hydrochloric acid. And you can see here that under this 1 molar condition, the naked gold aggregates, whereas the naked gold in water follows this profile here, and the Innova coat particles under all 4 conditions also remain well dispersed in the solution. We also subject our Innova coat gold particles to more serious conditions. Here we have 2.5 molar NaOH at 70 degrees. We've also done 10% DMSO at 95 degrees. And as you can see, most gold colloids aggregate immediately. I mean, most of these we can't actually get to the hot block before they aggregate. With the Innova coat, we're seeing nearly 2 hours, 3 hours, depending on the stage at which that particle is, whether it's coated and what it's actually conjugated to. So the significant increase in the stability of that colloid. This is an example of the Innova coat biotin gold versus a ligand self-assembly biotin-related gold. And you can see here we've got a streptavidin spot on the nitrocellulose versus a control with no spot. When we send the biotin gold up this nitrocellulose, you can see it binds to this spot very cleanly and there's no background. Whereas with the self-assembly, where that's dissociated or was destabilized initially, you can see the aggregation and the purple color of the gold even before it's reached the spot. And even on the control, we get this significant aggregation here. This can, to some extent, be reduced by optimizing your buffers, but under these conditions, you can clearly see quite a difference. Furthermore, when we do this sequential testing, so this is a demonstration of the integrity of the coat, A, B, C, and D are the same strip run one after the other. So we first ran the Innova coat gold up the strip and then washed it again. As you can see, there's no background there. And then we ran a naked gold at the same OD up that same strip, and you can see the binding to the protein IgG spot there very clearly. And even washing doesn't remove this aggregated gold particle from the strip. So this is the Innova coat gold. It's a proprietary surface coat which has provided ultra stability and also enhanced sensitivity. I'll demonstrate some data on that shortly. The covalent linkage of antibodies and analytes is quite unique in the industry, and the simplicity of the process also makes it extremely easy to use for proof of principle, early stage R&D, and is fully scalable up to manufacturing quantities. So we can discuss that further. If you have any questions, please email those through. So the principle is based on the same as the lightning link. With this very easy to use method, you simply add your antibody to the lyophilized particles, incubate, and it's ready to use. There's 100% recovery. It's optimized in such a way that you vary the amount of antibody that you add, and the efficiency of the reaction is such that you should have none or very minimal unbound material in there. Again, as we say, we've covered the stability and the sensitivity. I will now show you on this graph here. So here we have a large protein, IgA, a comparison between passively conjugated gold particles, all the same gold, the same antibodies, the same buffers. Everything was maintained exactly the same except for the passive conjugation here and the Innova coat conjugation here. This was read on a QIAGEN reader, so it's an arbitrary signal intensity, but you can quite clearly see the increase in the sensitivity between the Innova coat and the passive. And also here the percentage binding curve also demonstrates that. On a small molecule, again, more difficult to work with, and we see this quite often with a wobble at the low end on the fringes of the sensitivity of the assay. With the Innova coat, we've managed to increase the limit of detection to a lower concentration here, and you get this nice smooth curve. Just to finish off, we have recently been awarded a grant by the Technology Strategy Board in the UK, and we're further developing both gold particles, latex particles, magnetic particles, and various others. So if you do have an interest or you'd like to speak to us about that, please don't hesitate to get in touch. I hope this has been useful to you.
Okay, thank you very much, Ahmed, Klaus, and Tom. Please keep an eye out on our social media channels as well, because we will be soon advertising our upcoming webinars. And if you would like either a PDF copy of the presentation or a link to the video recording, please get in touch. And thank you very much for joining us today for our Beginner's Guide to Lateral Flow Assay Development, and we hope that you enjoyed it.